Thank you so much, Maggie, for this very thorough introduction. And I'm so glad to be here. Thank you for hosting me. And I've heard so much about a UVM Complex Systems Initiative and Center, and now Spire, and knowing also Josh quite well, and Stuart Kaufman, who was the keynote speaker at GDS last summer at Gecko. I'm very glad he could make it, at least via Skype. It was fantastic. So it had been a long time wish of mine to come here. I'm really excited to be here, and the schedule that has been put together is also fantastic. I get to meet so many of you colleagues from complex systems and talk about various things. It's great. It's just great. So I'm going now to bother you a little bit with my own stuff for less than 1 hour, I hope. And I'm very glad to announce that I ripped off the first 12 slides about complex systems from this presentation, because I'm not going to tell you what complex systems are. Something I need to do with other audiences, especially when I give this talk in computer science. So I need to go through that, and I end up having less time for the real interesting stuff later on. So you'll be happy I won't impose this on you. But what I want to focus on is the intersection of complex systems, the complex systems community as we know it today, even though, of course, internally it's very complex and ill-defined in many respects, but still there is some consensus about what we are all interested in. And engineering. Can we have an intersection, a working, functional intersection between self-organization on the one hand, those wonderful phenomena of large sets of elements interacting locally, emergence, and so on, and some concept of design, some concept of engineering, some concept of those elements making together an architecture, a functional architecture, not just random flux, random stripes, random clusters of nodes in a network, but something much more elaborate than that, much more architectured and spontaneously. We see that there are architectures without architect around us. Right, we all believe that there is no architect to them here. And so can we being fascinated by this, and I mean by that morphogenesis, biological development on the one hand, also termite mounds on the other hand is another big example. Looking at them, we think, oh, can we do this actually artificially, and or can we tamper with existing complex systems? A dangerous idea, sounds ominous, but this is what synthetic biology wants to do. Can we tamper with existing complex systems to make them do different stuff they are not used to doing? So I'm going to travel back and forth between biomodeling, understanding what's going on, bio-inspired engineering, importing what we have understood into artificial stuff, and then back to bioengineering, re-importing that bio-inspiration we had, and back to the source of inspiration, to the matter, and tampering with matter, right? So kind of back and forth, especially in the biological domain, which is the most exemplary of this intersection between architecture and self-organization, right? And so I've contributed to other lines of work in spatial evolution, a bit of ecology, artificial ecology, even a little bit on social networks, but little of that. So it's not my strong point. And of course, having been director of this institute in Paris, I had many colleagues in all those domains of complex systems. But now my own work is there. All right, so let's move on. These are a few vignettes of my work I'm going to present. So among all these complex systems here, this is the touristic brochure you can find at the Complex Systems Hotel to show you what's going on. So you can visit the molecules giving rise to cells, and the cells giving rise to organisms and their nervous systems, the organisms giving rise to those fantastic collective phenomena, and of course this organism here with its artifact giving rise to all those major societal issues that our policymakers are struggling with. All right, fine, but in that brochure, I want to focus on that. I want to focus on those complex systems who exhibit architecturedness, if that makes sense. I have never measured it. I have asked, in fact, information theorists and complexity measure guys, is there such a concept as architecturedness? Can you measure that? A system is not just complex, but also has something that really resembles, a plan, a design, and so on, but dangerous waters there, of course, except they are, of course, completely spontaneously appearing over evolution, over development. So maybe I must admit, you know, a bit the same interrogation as some of those creationists have, except, of course, with radically opposite conclusions, showing that you can have the illusion of architecture, and yet it has spontaneously appeared, completely spontaneously, I insist. So, and this is my goal, precisely, and can we do the same in technology? So, biology strikingly demonstrates the possibility of combining pure self-organization and an elaborate architecture, and there is a whole class of morphological self-dissimilar systems in which morphogenesis is much more than just pattern formation, and Darwin is thought of, I don't know if it was ever confirmed, but Turing, so many genius. So Turing has said, after his famous 1952 paper on morphogenesis, his last brilliant contribution, that he got the patterns right. He got, you know, the idea of how you get spontaneous appearance of order, amplification of fluctuations, and you got this striping out of nowhere, but he didn't get the horse that's around it. So morphogenesis is not pattern formation, right? And we have had a tendency to forget that. All right, so I want to focus on this kind of stuff, and especially that stuff here, this little guy and everything that it represents. Let me clarify. So there will be a few hot air slides, and then I will get to the real models after that. Let me clarify what I mean with more morphogenesis. So something like this. I'm very waving my hands. Some more and more structure and architecture. We start with famous ripples and random patterning. There is no architecture here. You do have very interesting texture, but no patterning, no architecture. Then you go into, for example, insect colonies, what they do. And here, you know, it's also a very diverse picture. You know, I would say that entrails are not that fascinating after all as such. Of course, they accomplish important functions, but also a network of entrails is also pretty much like this. But then when you get to the nests, it becomes really interesting here, because you have naturally much more architecture in the nest. And then even among the nest, I would argue that the termite mounds are probably the most amazing ones, because otherwise ants and wasps and so on and bees, it tends to be repetitive. Okay, of course, I'm simplifying greatly. This is for the sake of the story, but I hope you follow my intuition here. Termite mound is extraordinary in its heterogeneity, you know, specific structures inside and vents and chambers and corridors and so on. And it's like an anatomy. It's like an anatomy. And finally, we get there. Okay, so that's what I mean with this gradient here. I'm repeating this story here. And here I show also a bit more what I would put in the bag of random, statistically uniform, texture-like complex systems that have no intrinsic architecture, and those who do. And I would argue also that maybe what's different between these and those is that the agents that make those things appear have more information, whatever that means, have more instructions. You know, they are heavily loaded with a richer repertoire of behaviors they can take. A grain of sand certainly cannot do many things besides just, you know, clumping with other grains of sand. A cell can do much more than a grain of sand. It's obvious, right? So maybe by endowing physics with information, we get to have these things, and conversely, we have our beloved artifacts here that are strongly architectured by us and have next to no self-organization in them. And in that case, there is a bit the opposite movement of looking at what happens when they become self-organized at a higher level. So anyway, I won't comment too much on that. This is just the broad picture of the book that's going to appear. The fact that morphogenetic engineering, this field that I launched with a few colleagues, in fact, Hiroki Sayama is one, a few years back. We started with workshops and so on, is there at this intersection between engineering and pure self-organization. Somewhere here, there is that. There is a book, shameless plug, 1 minute, 20 chapters of various people, all contributing more or less, maybe without being aware that they were doing morphogenetic engineering, but we told them, hey guys, you are in fact doing morphogenetic engineering. So they said, oh, that's great. And so, so they contributed a chapter. And so we have a lot, of course, of works like Josh, naturally, and And back then, I didn't know Josh very well, so he should have been in the book anyway, but of course, this is similar works, Marco Dorigo and other people, Justin Warfel. And so you have this virtual or more or less real collective robotics, but Evo, Devo, morphogenetic robotics, Yao Chu Jin and people like that, Alan Winfield. So where you want your little robots or virtual agents to form something architectured. Again, you also have the more fluid kind of work here with Hiroki Sayama's swarm chemistry. You have heard of it probably. Then you have the more biological inspired stuff, my stuff here, I will talk about it, and Jake Beal here, and other people, Sylvain Cussat-Blanc, who stayed with Jordan Pollack recently. And then you also have the generative guys here. So this is just a sample, of course. The book couldn't have 500 chapters, but we find a bit the same crowd as in the GDS track, second plug, which will be happening next summer and co-organized with Josh this time. All right, so moving on and getting to the real stuff in 1 or 2 slides now. How do I manage to be long even with just a few slides? I don't know. So complex systems science and complex systems engineering. We are looking here at, we look back and look at those attempts at trying to link both sides, right? It has happened before. It looks like it has happened before. So people have looked at neurons, at ants, at genes, and then, you know, created those new disciplines that we find so interesting, neural networks, SEO, GAs, and so on. So, you know, this is, you can argue that this is somehow complex systems engineering, although you could also argue that those fields, or most of the works in those fields that are now traditional and classical, have strayed away from, you know, emergence and collective self-organization of little units. We don't see much of that going on anymore, certainly not in machine learning. You have lost that almost completely there. And so I would like to, and let me skip that for now, I would like to explore another avenue that hasn't been very much explored so far, from, instead of ants, genes, or neurons, from cells of a multicellular organism getting together. This is one of the wonderful videos that Nadine Perrieras, my colleague, my biological developmental colleague at CNRS, is producing, and she's co-supervising a thesis I will talk about. Let's look at this black box of the modern synthesis that has been a bit, that has accumulated dust over the past century, and now is being reopened by Evo Devo biologists, and I hope to be also contributing to reopening it in the artificial world too. So can we also look at opening the black box of development, aka the fine-grained complex system structure of one individual? So instead of thinking of individual as a dot, genotype, phenotype, it's all the same. Let's zoom in on the individual and realize that it is made of a lot of little things inside. And I will tell you why I want to do this. Anyway, so that's my endeavor here. And there is also the looping back. I will show, as I said, biomodeling, bioengineering, bioinspired engineering, bioengineering, looping back there and changing things. And this is why I want to do this. This is only speculation, as I think that the fine grain is really important if you want to see new structures evolve, appear, because they are made of these fine grain blocks. This is what authors in Evo Devo argue, but I will skip the concept and really get to straight to the matter. I will show here, if I have time, on this washed out projector, I will show 4 studies, or at least 3, and then maybe the 4th one too, that illustrate this movement between science and engineering I was talking about. A model of embryogenesis by my student Julien Delille in Paris, co-supervised by Nadine Perrieras, trying to be as faithful as possible to the early stages of zebrafish development in 3D. Then I will show now purely artificial stuff, physics engine world, 3D creatures walking and kicking balls and climbing stairs. So fine-grained Evo Devo robotics, or in fact fine-grained Devo robotics so far. The Evo side is still very thin. And then back to synthetic biology, a few slides about what we plan on doing. We haven't really done yet, but the idea there. And then maybe a little bit about self-constructed network, but I might skip that by lack of time. All right, so let's start with the biology here. And given the time constraints, I, of course, cannot say a lot about all the details which are very, very numerous. But here is in one shot Julien's thesis. It's a simplified view of his methodology and workflow. So Julien Delille with Nadine Pereiras. Here are the real images, the real imaging from the microscope. So we have 3D plus time, 4D movies of gray levels or variously colored embryos, but we don't use so far specific, in situ hybridization or specific marking or specific expression. It's just, maybe we can distinguish with some clever optical methods membranes from nuclei, but there is no differentiation of the cells that we can see. Anyway, take this. This is unstructured data. These are voxels when you reconstruct them in 3D, and create out of there a structured data, right? Here you are tracking. This is pure image processing. There is no model here. You just try to follow all the cells here that proliferate. This doesn't correspond to that, of course, but so you follow the cells and their flow here. This is a more advanced stage. And you segment the nuclei, the membranes. You look at the optical flow and so on. You process those images and you want to extract structured data from them, basically a long list of X, Y, Z coordinates, right? Okay, in time. And then this is a very vague arrow here, because here is your brain. You look at that, you say, wow, and you think, how does that work? You read the literature, you try not to be completely wild, and you understand a bit what biologists have figured out, but also it's extremely difficult, especially the mechanical aspect of cells is baffling, absolutely baffling. What the heck are they doing? Sure, they polymerize cytoskeleton and adhesion molecules and so on, and depolymerize them on the other end, and so that's how they kind of progress and reshape and migrate and so on. Who is pushing who is pulling who in this big crowd movement? Causality is a big question that biologists are fighting around. Why is epibody happening? Why is internalization happening? Who is leading? Maybe no one is leading, but then why are they doing it? And so on. So, sorry, I was here now. So you need to think about some model, and you cannot also be completely emergent, like at the molecular level. You have to go straight for the phenomenal level of, okay, cells are, it's as if cells were disks, and as if they had forces, like elastic forces between them, and so on. All right, so this is a vignette I will explain. And then you take that, and you run it, okay? You take that and you run it here, and you obtain a completely calculated embryo, okay? So this is really a pure simulation here with hand intervention. Of course, the differentiation, for example, in that case is a bit staged and scheduled by hand. You have to tweak by hand because the process is so heterogeneous in time and space that you have, in that case, you cannot expect everything to emerge, right? There is no GRN inside the cells at this point. There should be, but there is none. Anyway, and then ultimately you take this, you take that, and you try to compare, and you try to say, okay, this looks like that. So I must be close to the real thing. You need to validate the agent approach, right? So this is what he does, and now a few slides showing the details, or a little more of the details. So first we have naturally the beautiful microscope movies, just to tell you we are just looking at the early stages. We are not here yet, far from it. So just the early stages that it's already very, very hairy and complicated. All right, and as I said, different. You can have also this a new generation. Collaborate with a lab in optics, and they have those incredible, harmonic generation technology that can already differentiate between different substances of the embryo without staining them in any way. But there is no way we can really see different types of cells yet. Anyway, moving on, then the incredible workflow of reconstructing image processing only, no model here. Image processing, a huge team of people, I was part of it, huge European project, many countries involved, lots of mathematical methods, level set and Hoff transform and all that, flying over the details, but it's all about, filtering, detecting the nuclei, segmenting, tracking, and so on. Incredible work that all these students did, and so we are here in the workflow here, right? So, and here is an example, superimposing the detection with the gray level voxels, all right, to show you, and And then, by the way, the coloring of the different cells is made by hand a posteriori. Okay, so again, I say we have no way to distinguish them with a microscope. So what they did here is that they saw the cells, and then the biologist annotated the cells by hand in the end, and then played the movie back. So the coloring is done by hand. The coloring is done by hand. All right, Julien, my PhD student, has a different workflow. I'm now flying over the different. This is a different workflow that he made himself, slightly less ambitious, but what he wants to get at from the images, and it was also a nightmare of, superimposing different views and so on, and I spare you all that, and we want to get to this, which is this hairy ball here of vectors of flow of cells. That's what he wanted to extract. In his case, in fact, he didn't really need to know exactly where the cells were exactly one by one. He was interested in the migration flows. That's what he wanted to get at, what he calls the blended deformation fields. And that's what he's going to compare his simulated embryo with, all right? So that's his workflow. Then here we go. Now the centerpiece. So centerpiece is the model. And I will be using this explanation here also for my stuff, my artificial stuff, I mean with another student, Carlos, because it's valid for both. It's valid both for biomodeling and bio-inspired developmental engineering. All right, at the heart of things, when we talk about embryogenesis, multicellular development, we talk about, as Enrico Cohen is this plant biologist in the UK. He wrote a fantastic popular book entitled The Art of Genes and compares embryos to pieces of art, sculptures and paintings, and says that the embryo is in fact self-sculpting and self-painting at the same time. There is, of course, a hidden geography on and in the embryo. You have these different domains of cell types that correspond to different colors, quote unquote. A color can symbolize for a cell type. And literally, you can see colors if you, again, mark your cells with chemical or radioactive markers of some sort. You can reveal, hey, those cells are expressing this gene, right? So the embryo is constantly self-painting while the canvas is deforming, and it's also self-sculpting. In fact, I don't know many examples of pieces of art that are painted sculptures, except maybe, I think in the medieval times they had painted statues. Nikit Saint-Phal in Paris is famous for that, but nothing that grows and self-does, okay? This is what the embryo does, and okay, now beyond the artistic metaphor, which is very useful, I think, what is behind it, the scientific side, is naturally mechanics and gene regulation. And both, of course, are coupled. This is all about coupling both. So this is what makes it especially difficult, especially there. A little less there, in fact, but mostly there. So differential adhesion gives rise to modification of cell size and shape, which gives rise to growth, division, apoptosis, and motility migration in some ways that are very difficult to understand. And of course, this will create new neighborhoods. Things move, so they change their neighborhood. Hence, new signals, new cues as what you should become. You were pushed there, or maybe you pulled people there, and then you are there, and you receive new signals. Oh, okay, I should turn now green, now I should turn dark green, and now I should turn dark olive green, maybe, and so on. I will sub-differentiate, and so on. All right, and so also new gradients, the whole idea of positional information, I will be insisting on that very much. Whatever its biological reality is it morphogen, is it signaling? Okay, but there is some sense of to know where you are, triggering new regulation of genes and naturally triggering new mechanical behaviors. And it's not just this, of course, it's a bit everywhere. So on the mechanical side, you have many ways of modeling that. So we are in the modeling section here. We're at the top of the, we have many ways of modeling that and many different scales of simplification and caricature, or scales of trying to be a little more accurate. You have tensegrity, Donald Ingber, you have cellular pots, James Glazier, you have, this is a polyhedron, never mind. And this is in fact the simplest way that my student, after exploring many ways, decided, okay, this is enough for his purposes. Cells will be dots and adhesion will be springs. Good, but even there it's already complicated. And then the gene part, about gene regulation, genes promote and repress each other by binding on the DNA at regulatory sites. And so in space, then, this is an illustration by Sean B. Carroll. In space, that creates intersection of painting domains, right? So genes overpaint on top of each other and create those smaller domains in the cascade of regulation of areas where you have specific new genes being expressed because this and that and those genes were like that. All right, so the geography of the embryo is controlled by GRNs, which are a simplification of reality themselves. All right, so now Julien in his work spent almost all his time on the mechanics part. There will be almost no gene regulation in his case. And then I will show something which has gene regulation. So even at the mechanical part, you can make very simple assumptions. Let's assume it's as if it were Newtonian. You know, you have those kind of fluid mosaic and soft bodies, but let's assume they are like disks and kind of have springs between them. It's horribly simplified, and yet it can give something interesting. So then he kind of design and control a potential of attraction, repulsion. So this is the passive aspect of trying to stay together. And then there is the active force of wanting to migrate. And so he also studied local migration behavior. Sorry, so many details. But anyway, this is the mechanical part. And you also have to solve the issue of neighborhoods. So maybe, you know, it looks disappointingly simple. And yet even there, when you have thousands of these obeying that, it's already extremely complex. So now results, you know, now we are here in the simulated part. We take the model that I briefly, you know, hovered above and explained the idea behind it, but didn't give much detail. But anyway, this is the main result. And again, this is not proved by video. We want to compare. I will show you how in the last slide. But we have a few migration studies here. And we have also here, he shows, he had to make ad hoc decisions. You know, he had, again, by hand to decide, okay, this is the yolk, by the way. And the yolk is not made of cells. The yolk is a cell or a volume. So the yolk, in its case, is modeled with triangular meshes. And then it has those cells on top that in that case, he wants to show the different predefined types of his model. There is the envelope, the EVL, the enveloping layer, and then you have the cells that here at the surface of the yolk, you know, hypoblast, epiblast, and so on. So now this is the main result here. And again, it's mostly the effect of letting the cells run with the mechanical model I showed, plus by hand intervention as who is becoming blue, who is becoming green. So you know the dorsoventral lateralization and things like that. So many details. I have no time to. This is an overview of this work. Maybe you can ask me questions later on. All right. So. And now, last but not least, this is not an exercise in this case. This is not a futile exercise in producing more or less colorful videos. It is eventually an attempt at modeling the real thing. So here is an example of how it's done. This is illustrating the methodology. This is a much simpler video as the previous one, but this is another example. This is more like we call it the chewing gum, I think. Anyway, so eventually what he wants to do and what he has done, in some cases, is take his hairy ball of extracted vectors from the real images and superimpose them on top of this, where, in fact, what he does, he applies the same image processing methods to this. He takes this as if this was a microscope video and also runs it through its image processing workflow to extract vector flows. We don't show them here, but we should. And so then these two sets of vector flows superimposed, and there is a simple idea of a distance fitness of some kind that will try to say, hey, this is behaving collectively very much like that. In that case, we superimpose them on the fake embryo, which is a bit confusing, sorry. So this is the real vector flow, and this is the fake embryo, which also has a fake vector flow, and then we look at them. And then there is a bit of this vaguely evolutionary idea, except in that case it's just sweeping. sweeping the space and modifying some parameter. In that case, I think it represents some timing where epibody is going to set in and is going to be allowed to go in. So you also have a bit of staging, I said. And it happens that this one is the best in terms of has the best overlap. You see the idea, the concept. This is what we are trying to do honestly, and it is naturally very difficult. But Julien has very interesting results, and he's going to defend in a few months. All right, so that was Julien and embryogenesis modeling. And now I have, let's see, 20 minutes left, it looks like, or a little less than that, for the artificial part, the bio-inspired engineering part. And then I will say a few words about, coming back to the biology and trying to organize its self-organization. All right, so what about that? So a few of you might have seen these older ideas here in 2D, but I will show newer 3D results, not just for the sake of 3D, but also for the sake of behavioral 3D in an environment. So getting a little more serious there, not just having those things on the screen. But anyway, I must start with that because it was my concept. Now it's been 6 years now. I started this after being inspired by popular books on development, I must say. Again, Enrico Cohen, Sean B. Carroll, and other people, and Louis Wolpert. And I was thinking, okay, how can we get a flocking, and this projector is a little washed out, but never mind. How can we get this flocking mass of cells to do something else than just blobs? And how can we have them move, position themselves, or divide where they should do that? And eventually come up with a reproducible body shape architecture. Again, the idea of organizing self-organized architecture, or getting architectural self-organization, whichever way you want to think about it. So, and I capture that. It was baby steps, the first kind of, and again, lots of ad hoc decisions were made, but never mind. I had this simulation that you can see here in one shot. This is the work in one shot. Then I will explain more precisely what's happening. But I start with a few cells. I needed to have sources of morphogen, so I have to start with them first ad hoc. Then I let them divide, and then key in that process that you cannot see very clearly here, but I will show it better later, is gradients. In my case, I'm not saying it's necessary, but in my case, I really rely on gradients. Gradients is a shortcut for positional information propagating from node to node, so it's very peer-to-peer, completely decentralized. You can call it gossiping, you can call it quorum sensing, maybe not, because it's really about a wave of differential information sweeping across the swarm and establishing counters in every cell. And it's very dynamic, and so if cell moves, they get another counter. This is, to me, absolutely the key to then be able to read out these gradients, a bit in the French flag spirit. Okay, fine, there is a gradient, and you read it out. Yes, it is a French flag after all. But here, I'm not just content with that. Cells move. Cells, there is a bit of swarming, there is noise in their position, so the gradient is not given. It's self-established first and foremost. And then those domains just stay red or blue. They will have a new behavior, mechanical behavior. Remember the loop between genetics and mechanics, right? Between sculpting and painting. That's what I'm trying to do here. By self-painting, they will have a new self-sculpting behavior. The pink ones and the whatever yellow ones they are going to now grow differently. Grow differently meaning divide faster in a direction that will depend on the gradients and so on. And then this is not over, because then inside those new domains, new mini gradients are going to come to self-establish. So you have local geography inside a bigger geography. And this is quite literally what Enrico Cohen is talking about in his book. So I should write to him one day. Anyway, never really contacted him. I want to invite him for GDS, in fact, but it was such an inspiration. So that was my crazy, idea of let's try it. And here is now how it works, a little in greater detail. So the mechanics is, again, ultra simple, same kind of mechanics as Julien in his attempt at modeling biology. In my artificial attempt, I also have springs and disks, and I have this kind of, elastic potential, whatever. So it's in fact the same potential as the flocking guys, Vicek and so on. They use the same for their boids, and so you have, of course, a hard core here, but then you have some attraction here, some repulsion here and some attraction there. And then if you go too far, you don't see each other anymore. You are off on your own. Okay, fine. So this is the mechanics plus division. Plus division. Here my birds divide in flight, of course, because there are cells. And they create a mesh of Delaunay neighborhood. I think it's the best neighborhood. It's a little computationally complicated, but this is really what it's about. Anyway, so here you can see that the gradient, okay, there is a singularity. There is this special cell that has a special role. Yes, there is. And I will tell you more about it. So it propagates a counter, so it has 0. And the rule in every cell is the same. It's completely homogeneous in the sense that there are 2 rules. If you have n, you give n plus 1 to whoever is around you. No direction. Everyone should receive n plus 1 from you. If you receive m, you should keep it only if you have more. So you keep the min of what you receive. That's it. 2 rules. Okay, a little more tweaking about avoiding plateaus and things like that. So there is a 3rd rule, but never mind. But in any case, it's extraordinarily robust. Just that. It's simple. Cells push each other. There is a little noise. They divide. They form this swarm. It's just a blob for now. But there is this robustness of this self-established coordinate system, or self-established coordinate axis for now. And there are several of them, I should point out. And this slide doesn't show them, but maybe this one, that's the 3D version. Sorry, I forgot the slide that shows other gradients. You need several, of course. Now, this slide here shows how the gradients are literally read out. And for now, my GRN, so the gradients come here. They are plugged in, so the gradients are the input values, the morphogens signaling whatever it is in this abstract world is going to activate the GRN in some ways and tell it to produce, to express these genes or that genes. In this case, I'm almost ashamed to show it. It's a perceptron. Okay, it's a feed-forward GRN. Morphogens in, 1 light bulb out. Okay, but this is terrible because GRNs are not like this. At a certain level of zoom, they are kind of a cascade. Drosophila, the cascade of the first developmental genes. You have some kind of cascade of genes going on. But of course, when you zoom in, it's much more recurrent than that. You have cliques of genes. We just discussed it with Josh. And so you certainly have attractor dynamics. I would love to replace this with attractors, constellation of genes, making the cells be green or blue. Anyway, but for now, it's that. It's fine. And this is why you get also a robust patterning, programmed patterning, not random patterning, not Turing patterning, okay? It is a programmed patterning coming from the GRN they contain. And so you have this scalable, and I could continue dividing and dividing. We would always get this kind of 3x3 checkered patterning as we go, okay? So this is the whole point. Now, here is the same idea in 3D. We are just talking about the body now. Just the body is a blob, is a disk or a sphere. Now, the new work with Carlos in 3D, with the ODE physics engine, same ideas. Really nothing new in that case. We also have this one gradient in 3D. And of course, we have several gradients. I didn't really show it very well. And you might wonder, where are the source cells for these gradients? Good question, thank you. And the source cells, in fact, self-organize too. They swim away from each other. They are not put by hand. We start with them, and then they kind of go away from each other because they have a rule telling them you should swim up the gradient of your opposite partner, right? So there is also self-organization of source cells. And then when we get to the local patches, the source cells become, in my case, they identify themselves. Oh, we should be source cells now for the yellow section here because we are at the borderline with the blue guys. So they detect that they have 2 different kinds of neighbors. Apparently, this is what happened in the imaginal disk of the Drosophila wing, or in fact, in the chicken's wing butt. There is this kind of Norwegian flag of local gradient arising inside this little patch, and that is triggered by border cells. And cells know that they are border because the cells near them are different type, and they communicate in some way. And so there is this idea here too that you don't need any longer to, of course, plant the gradient sources that might be a concern you have right now We don't plan the gradient sources by hand, of course. They then pop up on their own to establish those coordinate systems. Anyway, so that's the body. And now let's get to the limbs. And here, Enrico Cohen tells us that it's like a fractal, self-dissimilar fractal process of the embryo refining its details at various scales. So we have the big domains. This is the Ur body plan, if you wish. Here be a face, okay? And then inside the face, here be an eye, here be an eye, here be a nose. Naturally, you take this with a grain of salt. It's illustrative, it's metaphorical, but hopefully still makes sense. And then, so inside the domains, as I said, new gradients arise and will subdivide, segment this domain into smaller domains, and here be inside the eye, you know, an iris and so on. Something like this. And then, of course, as I said, the new domains have new mechanical behavior. For example, here, a meristematic kind of growth, happily mixing plants and animals. So, for example, the cells at the tip, they know that they're at the tip because the gradients tell them that they're at the tip, will grow more than the cells in the middle and so on. So you can now play. You have so many possibilities to play. And this was handmade design of the genome, right? That gave rise to this. And then you obtain a genome that is, in fact, modular. In that case, again, a feed-forward caricature of a genome. Modular and reflects somehow the modularity of this. But I will talk more about this in a few slides. Now in 3D, same idea. Same idea there. In fact, here we have fields of division. We have division fields as vectors, so some cells are not dividing anymore, and some other cells here at the tip of the limbs are dividing, very much like the one I showed you in 2D, the meristematic kind of growth, where the tip grows but not the rest. And why do we have these fields? Because they are a function of the gradients. Okay, so the gradients, again, are running inside, establishing those counters, and then cells look at this and say, okay, I should have a vector like that, all right? So fields of division as a function of the gradient can give rise to differentiated growth, and so new structures, all right? And so here is the full thing here. So this is the growth of the whole organism. I'm speeding it up. So at one point, and you might also wonder, when does the body cease to divide and decide it's time to grow the limbs? Good question too, and the answer is this is staged. This is staged, wait a minute, it's kind of half staged. So the limit of the body size is in fact in the gradients. There is a rule that says if you get a gradient 20, you should start screaming and say, hey, everyone, stop dividing. I'm at 20 here. We reached the body size. So you send a signal to everyone. And then it means that at that point, now everyone stop executing the body plan and start executing the limb plan. All right? So there is a bit of that discrete, arbitrary cutoff, body stop, now limbs. It's not like that naturally in the real thing, but never mind here. So we have a bit of a little bit of ad hoc intervention. I'm telling you that honestly, but very little. In any case, so this is what gives rise to that. Fine. Moving on now. Why doing all that? Why did we bother? Why did we bother with this mess of spheres and edges, and the poor ODE physics engine is on its knees and has never seen that before and is panting and it's slow? Why? Because it's fun, and because, here you go. And why is Microsoft doing this to me? No, it's good, okay. Because then here, finally, after my little 2D experiments, we are able to immerse it in a 3D environment with some physics. No, a gravitational field, just that, and a hard floor, a rigid floor. And here you go. Then we can start really, so this is embodied robotics, like Josh talks about. We can, now we have all this effort put in designing or letting evolve, I hope, later on, a genome that is going to create such a complex creature because most of the information or the abilities that the creature has is in its morphology, is there in its body. And I didn't talk about the bones and the muscles, but we also have, this slide is also supposed to, at the same time, remind me to tell you that we also have bone differentiation and muscle differentiation. So why do those cells know that they are bone and they should have, and they are colored in white? Because they are somewhat, for example, in the middle axis, the central axis of the limbs. And why do they know they're in the central axis? Because the gradients, west and east collide, and so the cells say, oh, I have an east gradient that is 6 and a west that is 7. I must be somewhere in the central axis. I'm going to be bone, all right? So that's how it happens here, although maybe in that case it's not exactly the central axis, but you get the idea. And so we have a skeleton. And so why is a skeleton here? The edges are given very, very high elastic coefficients. Almost infinity, but not quite. Anyway, and we also have muscles, and muscles are not everywhere. Muscles are going to be here at the base or the root of the limbs, and they are going to be the one who contract. Okay, so differentiation into limbs, into bones, into muscles, and put that in an environment, and then we had fun testing the skeleton. So we dissolve the flesh of the poor thing and just see, it's now actually actively being submitted to gravitational forces and resisting them, as opposed to the other poor guinea pig here. We stripped their skeleton off, and it kind of squashed on the floor and couldn't really, stay standing. So it's a proof that, for example, our skeleton, proved by video, that our skeleton is useful. And here we go. Now this is animated here. Now this is not animated. This is ballistic. This is just passive. Okay, just stay where you are and try to survive. Here it's animated in the sense that we also add the brain part. And the brain part is, in this case, very primitive. There is no brain, in fact. It's just the ability that other authors have done. We are not the only ones. My Polish colleagues, Boris Robol and Michal Joachimczak, and then Lisa Schramm with Yao Chu Jin. A few people have already explored twitching, and naturally Josh and Josh's work, where they also twitch, but they have a really evolved neural control of this. We don't. We simply say from the morphology, the muscle cells that are slightly pinker than these ones, if you can see, are going to be the ones that twitch. Okay, and so the schedule, the animation, the neural schedule is a bit ad hoc here. It's the same standard. We didn't really explore very much sophisticated neural control because we were focusing here on the morphology, on the structural aspect. And so you can apply a standard animation and it will give different things. And that's what I want to show now. Let's see. I'll talk about evolution later. And I have 1 or 2 minutes left, so I'll be fast. Here is a fitness, for a stupid fitness function, just how far does it go, for example. But we noticed, I want to tell you about the problems we have. We noticed that even though we had achieved very nice shape reproducibility, so when we run again the development, we get the same four-legged creature. By the way, the genome was designed by hand in that case, four-legged creature. However, interestingly, the behavior is very chaotic so far, and it looks like the exact, which we don't want this, but unfortunately, the exact position of the muscle cells and the foot cells make the thing rather sensitive to where it's going to go and what it's going to do. And because of that, we decided to bring the creature to the blacksmith and fit it horseshoes here. And in fact, also before that, give the tips of the limb some horns, so they're more rigid. And so we wanted to get rid of those, maybe those accidents of the fine grain, yet not enough fine grain of the legs on the surface that make it maybe tumble here and there. Or, so we got better results when we had rigidified the tips of the legs and fitted horseshoes. It was a little more consistent in its ability to always do something or not do something. And also, we thought that the rigid floor was really boring and not, we couldn't really do anything with it. So let's have stairs and let's challenge the creature to climb those stairs with the new hooves and horseshoes it had and see how far it could go. And so some fitness like that, how far it can go. And we were, we have fresh results. It's all, it's at the same time a little disappointing and at the same time encouraging. We have, some evidence of that some developmental genomes are better than others at this or that task, in that case, climbing the stairs. So in the sense that the limb size, and by the way, what does size mean? We don't have a genome of a blueprint, right? We have a genome of development. The size here is, for example, the gradient at which limbs stop growing. That's the gradient that is in every cell. Every cell knows that if they get 12, they should stop growing. And it looks like 12 is better, is somewhat an optimal value here. And the body size, it looks like fatter guys are better here in that case. So there is this tendency, but okay, this is so difficult to harness at this level of granularity that again, this is not really the ultimate goal. We don't just want to show that longer limbs or bigger bodies are more able. We would like preferentially to show that different structures are going to be better. This is what we want to show, right? And we haven't gotten yet. We haven't gotten there yet. This is my older attempt in 2D at least showing by hand again, that my creatures could exhibit different architectures, so qualitative mutations instead of quantitative mutations. Some biologists are going to shudder at that sound of maybe micro versus macro evolution, but wait, no, so levels of changes in the body, especially similar to the famous antennapedia, the famous idea that maybe if you tweak one regulatory link somewhere or a few, you are going to get something radically new. You know, a leg is growing where an antenna should have been, or is growing where it shouldn't have grown. Ectopic growth, ectopic growth. Ectopic growth giving rise to homology, you know, digits, segments of insects, you know, being duplicated again and again, vertebrae, teeth, and so on. Probably evolution also works like this. You have duplication of modules, parts, organs, appendages, and then because they are duplicated in a genome here, they can evolve independently, be mutated independently in different ways. So then once you have 3 legs instead of 2, some of them can get shorter, some of them bigger. Something like this. This is illustrative of teeth and vertebrae, for example. So this is what we want to get at in the 3D behavioral thing that we haven't done yet, all right, but which I have explored here without behavior. If you see where I am now, that's it. Okay, so, and finally, maybe I overuse, okay, 1 minute to synthetic biology, just the intention there is to apply this morphogenetic engineering principle to BioWare. So at the bottom here, this is the view that computer scientists have of synthetic biology. It's about compiling. Because synthetic biology is wonderful, you can make bacteria do different things, but if it comes to designing genomes and designing GRNs, you will never be able to do it by hand. You will never be able to go to the BioBrick catalog and say, I want this brick and that brick and that brick by hand. You will never do that. You need something automated to create those GRNs, which are going to involve thousands of genes. And this is where computer scientists say, aha, this reminds us of compilation. Let's have a high-level description of what we want and compile it down to the BioWare and let the BioBricks, you know, automatically being ordered and assembled. And this is where my endeavor with a few colleagues from this French project, Symbiotic, is to apply shape engineering ultimately to bacteria. We will never get there, of course, but we at least want to show that this is something. And Jake Bill, I must say, Jake Bill, who was from MIT and is at BBN Technologies these days, spatial computing guy, fantastic researcher, also has started this bit of a tower going down to synthetic biology of compiling down specifications. But he hasn't done morphogenetic engineering as such yet. And this is what we would like to do. So for example, how we study dynamics of GRNs, and we hope that one day we can plant a seed and grow this. This is a fantastic dream by Susan Stepney. Susan Stepney at York in the UK, a big promoter of unconventional computing, natural computing. Natural computing is about computing in material, right? Can we make the bacteria do things like that one day? It sounds completely crazy, but why not? You know, maybe or something more modest. But the idea of organizing the matter in more complex ways, and we have started doing this with my student, my postdoc collaborator, Taras Kovaliv, who has collaborated with Wolfgang Banzaf and John McCormack and people like that, using this very clever language, GRO, G-R-O, by Eric Clavins at U Washington. somewhere here. And so you can, the mechanics is given, you know, you are offered this bacterial platform, and then you can program them in specific ways to get not just a French flag, but a Korean flag. No, I mean, so things are moving around, and playing with this and hoping we can plug this to what I showed you earlier, the EvoDevo endeavor, maybe one day in bacteria, and acknowledgements are here. Thank you for your patience. I will skip that. No time. Here, acknowledgements, Julien, Embryo, Carlos, this thing, Taras, that, but also involved with co-supervising this. And I didn't show you these guys, never mind. Thank you.